Hello, students. I'm Dr. Arpita. Welcome back to our developmental biology lecture series. As discussed in the previous lecture, as I've told you that I have divided this lecture into two parts: sex determination in mammals in lesson one and in Drosophila in lesson two. In this lecture, we will talk about sex determination in Drosophila. If you have not seen the previous one, please look into the description box. I'll share the link there. Please do follow that video as well. Okay, so let's get started. From your exam point of view, sex determination of Drosophila is very, very important. So please be attentive throughout the lecture. Okay, so sex determination in Drosophila is achieved by a balance. It is achieved by a balance of female determinants on X chromosome and male determinants on autosomes. Please keep in mind here autosomes play a very important role. The femaleness is controlled by X chromosomes and maleness is controlled by autosomes here. So, what did I say? It is achieved by a balance of female determinants on the X chromosome and male determinants on the autosomes. Okay, normally flies have either one or two X chromosomes and two sets of autosomes. If there is but one X chromosome in a diploid cell, that is, if this is the ratio of X and A, that is autosome, the fly is male. Okay, see, the number of X chromosomes is to the sets of autosomes, that is, ratio of X and A, the ratio between X and A determines sex in Drosophila, right? Now we'll talk about chromosomal constitution in Drosophila. What happens here, you see, normal male flies have three pairs of autosomes and two sex chromosomes, that is X and Y. Clear? Females have three pairs of autosomes and two sets of chromosomes, that is X and Y. Okay, females have three autosomes, that is three pairs of autosomes, and two sex chromosomes, that is X and X. And what about this normal male flies? They have three pairs of autosomes like the females and two sex chromosome X and Y. Clear? Okay. Now see, this is the you must be thinking, what is this blank table doing here? I'll tabulate the data in a concise manner in this table here, so that you don't have to read pages after pages, paragraphs after paragraphs, which becomes very, very boring and tedious during your exams. Just look into this table and keep these points ready at your fingertip. Okay, chalo, let's start. Fine. You choose a nice color to write. Huh? Something that is nicely visible. Yes, this is nicely visible, but this is created as we'll rub it off. Fine. Fine. So we'll make the table first. Fine. In this we'll write X chromosome. In this one we'll write autosomes. Clear? This one we will write X is to A ratio, and in the last one we'll see the sex of the organism. Now, condition number 1. Serial numbers of conditions here. Okay, here in condition number 1, we have 3 X chromosomes. Okay, 2 autosomes. What is the ratio? 3 by 2, which is 1.5. What is the sex here? Meta female. Fine. Okay. Next, if there are 4 X chromosomes and 3 autosomes, then what is the ratio? 1.33. This is also a meta female. Now see, 4 X chromosomes, 4 autosomes. What is the ratio? Ratio is 1. Right? This is a normal female. So a normal female has X chromosomes and autosomes in the ratio 1:1. Right? This is all about female. Now let's see what happens in male. If there are 2 X chromosomes and 3 autosomes, what will happen? 2 by 3. This means 0.66. This is intersex. All right. Here you see next condition, 1 X chromosome, 2 autosomes. What will happen? What is the ratio? 1 by 2, which is 0.5. Huh? This is a normal male. This is a normal male. So you remember, normal female has a ratio of, remember this, normal female has a ratio of 1 and normal male has a ratio of 1 is 0.5. Fine. Anything greater than 1 in females will result into a meta female, and anything say greater than 0.5 will result into an intersex organism, intersex Drosophila fly. And the last condition, see, last condition for males here, you see, 1 X chromosome and 3 autosomes. What will happen? This is right. This is a meta male. See, if one thing I didn't mention for the females, if there are 2 X chromosomes and 2 autosomes, like 4 X chromosomes and 4 autosomes, same here, if the number is just less, but the ratio is again 1, then it's a normal female. Okay. So this table has to be understood and kept in mind. Apart from this, we should know a little about. So from this table, what information did you infer? One thing you must know. In flies, unlike humans, unlike mammals, Y chromosome is not involved in sex determination. Clear? You should know this. Fine. Next, what other things we should know, which is very, very important? They are genes responsible for sex determination. What are they? One is sex lethal. Sex lethal. This gene needs to be inactivated in male. This is the master gene, okay? This needs to be inactivated for females to develop. Next is the transformer gene or TRA gene. Transformer gene or TRA gene. Transformer gene. Third is what? Transformer 2. TRA2 gene. This is transformer 2 gene. And the last one is DSX or double sex. D double sex or DSX gene. You must know the names of these. These are the 4 important genes responsible for sex determination. Fine. See, in Y here doesn't take part in sex determination, but activate those genes that promote sperm. This one you should know. What did I tell you here? Y chromosome is not involved in sex determination. That's true. But it activates. This one I'll write for you. But it activates those genes that produces sperm. Okay. This one you will have to know. That means what? It is not involved directly, but of course it is involved indirectly. See this one, if I write here, you will understand. In XX flies, XX fly means what? Female. Here SXL tra tra2. This is female DSX and it will form a female. Correct? Why is it looking like this? Yes or no? Yes. Now for this one, you see XY means what? Male. SXL tri-mRNA. It will give a non-functional protein. This means what? A non-functional protein means what? A male. Clear? So this is what is happening in Drosophila sex determination. Hmm. See this in female functional proteins like DSX, TRA, TRA2, SXL is very, very important. These proteins will have to be functional in order to get a female, right? And remember for female formation, X is to A ratio also has to be high. These two names, I'm not sure whether you have heard or not. Numerator, I mean, in the X, hmm, X is that numerator protein, and that A is the denominator protein, which is dead pan and extra macrokete that forms the male, and cystoless A and cystoless B in the numerator, that is X, it forms the female. Okay. These little names here and there you must know so that you don't get confused anyway. Hmm? You will not get confused at any given situation. Fine. See, if this X is to A ratio is low, then what will happen? A will encode the denominator protein, which I just now told you, dead pan and extra macrokete. Hmm? And these will act as a repressor, and it will block the transcription of SXL gene. Clear? So you will have to be extremely alert about what type of question is being asked. Don't get confused. Read the questions twice, thrice before answering. Hmm? Fine. So this is our discussion on sex determination in Drosophila. This table has to be remembered at any cost. Hmm? Remember male and remember the normal condition, then you will not forget the others. Fine. Normal condition means what? See, for normal female value is 1, and for normal male it is 0.5. Right? Okay. So thank you so much for attending the lecture. We'll soon be back with another interesting yet important topic from our DevBio syllabus. Till then, keep learning, and above all, take good care of your health so that you are always fit and energetic to take up a new lesson and be in a good state of mind before your exam. Till then, thank you.